Hello, everyone, and welcome to the latest edition of our Lightning interview series. As always, we want to make these sessions interesting and interactive, so please do introduce yourself, tell us where you're joining from and what you do, and of course, please feel free to add your questions to this conversation. Now, as you well know, today's guest is Dr. Daphne Koller, who is a trailblazer in the fields of AI, machine learning, education, and biology, and also, I must say, a visionary and inspiring entrepreneur. But first, a few words about upcoming items. Now, it is a very busy month for us here at ODSC. We do have our ODSC East coming up in our hometown of Boston on May 9th to 11th. And we also have ODSC Europe on June 14th and 15th in the great city of London. And lots of good stuff going on there. As you probably have noticed, the full schedule for ODSC is live. And we have a wonderful lineup of some of the best and brightest speakers, keynote speakers, and instructors in AI presenting that conference. And it will be both in person and virtual. So do check out our schedule if you haven't already. And yeah, lots of fun things going on. We're working with VV8, and OpenAI. We're going to be doing a generative AI hackathon there. We'll have cash prizes, lots of cool swag, and I believe we'll have some access to some early access to some of the plugins going on there. And of course, as one of the community things we do, we will have a career fair there and an education expo. So if you're looking for a job, I think we've got a number of companies hiring from across industry and government, as well as some fantastic universities, including some of our neighbors here, Northeastern Boston College, Merrimack College. And and if you're looking to hire in this space, looking for people, please come along. We're going to have a job board. And again, it's all free, so check that out. And I think we have a virtual version of that as well. And have to say, as you may know, Boston is known for being a hub for biotech and life science. So it's very fitting that in addition to hosting about a dozen tracks across machine learning, deep learning, data engineering, NLP, LLMs, we are hosting second year in a row a track on AI and biotech and machine learning track. And it's quite fitting that today's guest will be our virtual keynote speaker giving a talk on transforming drug discovery using biology. And of course, you have access to that keynote and all their keynotes for free on our website, so you can go check that out. And that's also a good segue into today's interview, because it is my immense pleasure and honor to introduce our, I have to say, esteemed guest for today, and that is Dr. Daphne Koller. And throughout Daphne's 18-year tenure at Stanford University, she has received numerous awards and honors, including the MacArthur Foundation Fellowship, the ACM Prize in Computing, the AAAI Alan Newell Award, and also a prolific author. She has published over 300 highly referred publications, and her impact is further exemplified by her inclusion in Time Magazine's 100 Most Influential People, as well as her induction into, I would say, some of the most prestigious societies out there, such as the National Academy of Engineering, the American Association for AI, the International Society for Computational Biology, and that's to name just a few. And as mentioned, Daphne is not only a trailblazer in the fields of AI and machine learning, but she is also a visionary entrepreneur and has worked tirelessly to make a significant impact in both education and healthcare. And I must say, as co-founder of Coursera, along with Andrew Ning, nobody has done more to democratize AI in other fields and providing high quality education for millions of learners around the globe. And today, she continues to push the boundaries of science and technology as founder of insitro, a company harnessing the power of machine learning to revolutionize drug discovery and development. Daphne, welcome.
Thank you. It's a pleasure to be here, Seamus.
And Daphne, I got to say, that is an incredible list of accomplishments.
Thank you.
So first question we have to ask is, tell us about your journey to becoming one of the foremost experts in the intersection of biology and AI, and really what motivated you to pursue expertise in both fields?
So my origins are actually in machine learning, and they date back to a time when machine learning and AI in general were, let's just say, marginalized in the field of computer science. I got my PhD working in this space at a time when you couldn't really say you were doing AI. You had to say you were doing cognitive computing, because if you're doing AI, you were kind of like a little bit out there, which is kind of funny when you think about where we are today. I was actually among the first people to enter into the field of machine learning from within AI. When I started in the field, many people were still doing logical theorem proving type stuff. And so I was the first machine learning hire into Stanford's computer science department all the way back in 1995. And I became interested in machine learning in the service of biomedical data, because way back then, the data sets that were available in core machine learning were actually not very exciting and not very inspirational. It was things like filtering email messages into spam and non-spam, or deciding a bunch of newsgroup messages which category they belonged in. And they were very small and not very aspirational. And this was around the time in the late 90s that biology was actually starting to measure itself in larger scale ways. So the first microarray data sets were coming out. The first human genome was coming out in the early 2000s. And I felt like there were incredible opportunities to do something that was both more societally beneficial and also more technologically interesting. And so in the first few years, and this was around the late 90s, the first few years that I was in that space, I was mostly focused on it as a technological challenge problem. But then I became interested in the field in its own right. And so throughout the majority of my Stanford career, I ended up actually having a bifurcated existence, where half my lab continued to do core machine learning research published in the standard computer science venues, ICML, NeurIPS, and so on. And the other half were published in journals like Nature and Science and Cell. And what was funny was that my computer science colleagues, most of them didn't realize I did biology. I still have people walking up to me today and say, "So what made you get into biology after Coursera?" And it was like, no, it actually happened like 20 some years ago. And the colleagues that I had in biology didn't realize I could be in a computer science department. They assumed I was in the medical school. So it was very interesting to see, to have that sort of real bifurcation of my research.
Yeah, incredibly impressive and a great inspiration for those who want to work in multiple fields. So you're, I know you're doing lots of things currently, but one of your main focuses is as a founder and head of insitro, which we mentioned in the introduction. Can you describe the core mission and goals of insitro?
So when you look at the current state of drug discovery, and unfortunately most of us, either ourselves or our loved ones, probably all of the above, will end up needing therapeutic intervention at some point in our lives. And you look at how long and how complicated it is to make a medicine for a new indication, and when you look at the statistics for that, you realize that even of drug programs that actually enter the clinic, 90% of them fail. What other industry has a 90% failure rate? And so the reason, I think, is because we are trying to intervene in a system that we just don't understand. We don't define diseases properly. We define them typically using coarse-grained clinical symptomology, very subjective. And then we try and do interventions in these really complex systems without truly understanding what's going on. I believe that the challenge is that the human brain just cannot understand human biology in its complexity. It's just a very challenging problem. And so we believe that the path forward in order to make meaningful therapeutic interventions is to collect as much data as we can about the human condition, about the human biology, about cell biology, and then put that into a machine and let it do what it currently does better than people can, which is understand subtle patterns, connect them to each other, and then use that to identify meaningful causal interventions that could truly modulate disease in a subset of humans. And so that's what we're trying to do, trying to collect a lot of data, trying to build machine learning methods capable of interrogating it, and using that to translate into therapeutic interventions.
Fascinating. So I do want to get into your comment about the failure rate of drug development being so high, but one quick comment, a credit to you. I think data-centric AI right now is all the buzz, but I think you had a data-centric startup quite a number of years ago. So when people didn't realize, or were starting to realize that the importance of data, now you see with large language models and anything else as well. So it's when we say you've been a visionary, it's not for no small reason. But for just stepping back a bit, for those of us not so familiar with the biotech industry, and we start to talk about AI and biotech, can you explain the key differences between traditional drug discovery approaches and in sitro's AI-driven methodology, which you just referred to?
So, and let me just say that what I'll be describing is a little bit of a caricature, because of course there's a multitude of researchers working in drug discovery, and there's a multitude of approaches that they take. So I'll be providing kind of like a little bit of an extreme version, but typically drug discovery emerges from a biological hypothesis that a scientist comes up with by reading papers, doing some experiments in small model systems, saying what happens if I do this, and you basically follow a hypothesis-driven interrogation, and ultimately the scientists and the powers that be in a pharma company develop enough conviction around a particular hypothesis, and they take it through into drug discovery. We take a very different approach that is anti-hypothesis driven. We collect as much data as we can about a particular type of biological system that we know is implicated in the disease process, and we go as unbiased as we possibly can. So, for example, when we collect data on cellular systems, we measure them with all sorts of different measurements, like the level of every possible gene in the transcriptome, like how active is it? We measure cell morphology. We measure cell trajectories as they evolve over time, as unbiased as we possibly can. And then we use machine learning to have the hypotheses emerge from the data and use orthogonal forms of data to gain conviction around those hypotheses. So in some sense, rather than starting out with a hypothesis and then doing experiments to test those out, we do experiments to feed into machine learning models who tell us what the interesting findings, the interesting insights that emerge from those data, and then we select among those which are the ones that are most likely to give rise to a meaningful therapeutic intervention.
No, extremely interesting. And I was just looking for a stat this morning, prepping for this interview and reading that the traditional approach to drug discovery, the average drug takes anywhere from 1 billion to 2.6 billion. And obviously, when you're dealing with averages and you're dealing with ranges, who knows what that means? But are you seeing that AI can have significant impact on bringing those costs down, or is it too early to say? Because I know there's lots of other things involved in the traditional approach to drug discovery.
There are many aspects that contribute to those costs, and we can start to tease apart the increasing costs of clinical trials and so on. But I would say that the biggest contributor to the cost by far is that we simply is that the journey of a drug from idea to approval has many forks in the road, and we generally don't know which one to take. And so we end up choosing the wrong one, which can lead to a long and costly detour, or sometimes even a dead end that kills the program altogether. And so the main contributor to that incredible cost is the fact that each successful drug carries on its back the cost of all of those failures. So we believe that machine learning, by making better predictions at multiple of those forks of which one to take, is going to eliminate, or at least considerably reduce, the number of failed detours that we take, and therefore reduce the costs and also reduce the timelines towards a successful outcome.
Yeah, and I just love the data-driven approach to it, because there's so much to be looking at here, because like everything, context is everything. So you have, I'm sure it varies by therapeutic area and which phase of the clinical trial, because some of the data I was looking at, just as a data scientist, was very interesting to me, that the phase 1 clinical trials, again, just dealing with averages and ranges, was quite high, 67%. It seemed to drop even further to something like 30% for phase 2.
Right.
And then maybe increase a little bit in phase 3.
Yeah.
I know the European methodology is a little different. And there's these different risks and different challenges at each stage. So have you found that in terms of AI and which are data sets that it works particularly well? Because you mentioned failure rates earlier. Do you find like it works particularly well in one area, like phase 1, phase 2, phase 3, or is it really dependent on again, well, I'm sure it is on the context and the therapeutic area and things like that?
Yeah, so let me break that apart for people who are not experts in clinical development. Phase 1 is invariably a safety trial. You put the molecule into a person in escalating dose, and you hope to avoid seeing toxicity or adverse side effects. And though we've become pretty good at this point as a community at making drugs that are safe. And so relatively few drugs fail at phase 1. Phase 2 is where you first test out the biological hypothesis that made you go after this target in the first place. And the reason why most drugs fail at phase 2 is because that is the first time when we realize that the target that we've selected to go after with a drug actually doesn't modulate the clinical outcome that we set out to modulate at all. And so that's really where, if you will, the rubber hits the road. Now, phase 3 is where you take the findings that you got from what is typically a lower powered phase 2 trial and really test out that hypothesis in a much larger population. It's also the time when some of those sort of rare events that cause toxicity come out because you put the drug in a much larger patient population, gives you more of an opportunity for sort of weird outlier people that interact in unexpectedly poor ways with the drug, that emerges. But the main cause for failure in phase 3, other than those unexpected outlier events, is people who squint at the data in phase 2 and say, I didn't see the signal I was hoping for, but you know what? I'm going to try it anyway. Yeah. So I think that's partly, honestly, a matter of just having the rigor and sort of willingness to say no to pushing things forward that are less likely to succeed. But phase 2 is really where the rubber hits the road. So what we do, we're not in phase 2 as of yet, or even phase 1, but the failure in phase 2 begins a lot earlier. It begins when you first select the target and the patient population in which you put your medicine. So the fundamental biological hypothesis that even incentivizes you to go and make a medicine against that particular target. And so a lot of our discovery work is really focused on how convinced are we that if we make this intervention at this target in a given population of humans, that will modulate this clinical outcome. So we call that a clinical hypothesis, and it's effectively making a prediction around a causal intervention. Because when you put a drug in a person, this is not an observational trial. This is a causal intervention, and so you can think of it as building a sufficiently predictive model of human biology that it can predict outcomes of causal interventions.
Wow, and so that sounds like both fascinating and highly challenging. So insitro started in 2015, I believe.
2018.
2018. So I'm sure you guys have addressed many challenges. And what would you say one of the main challenges have been in integrating AI with experimental biology? And how did yourself and your team address those challenges?
So I think if you go back to the statement I made a moment ago, which is that we're looking to build predictive models of causal interventions in humans, and we're doing it using machine learning, the first question that you ask yourself is, we don't get to make causal interventions in humans very often. When we do, it's called a clinical trial, and by that point, it's too late. So how do you get enough data around causal interventions in humans in order to build predictive models? And in order to do that, we've taken a rather unusual strategy of bringing together and bridging between two very distinct and complementary data modalities. The first of those is data from humans. And as I said, you don't have a lot of ability to interrogate causality in humans because human trajectories are invariably confounded. With non-causal associations. But one very important ground truth that we have around causality is human genetics. So when my genes are go in one direction or another, when there's a particular variant in my genome, it causes a certain phenotypic trait downstream. I mean, it's not perfect. There's often a combination of multiple genetic and environmental factors, but it is one of those few cases where you are pretty certain that the causality goes in one direction and not the other. And so we use data from human genetics and complex phenotypic traits that are measured in humans, and that's exactly to avoid the hypothesis-driven sort of looking under the lamppost of we know we believe we know what matters because a clinician told us what matters. We deliberately put that aside and say we're going to measure human biology in unbiased ways that emerge from high content modalities such as blood biomarkers, histopathology, MRI imaging of the brain or the body. I mean, lots of high content data, and then relating that to the genetics as a causal root of this to understand the effect between genotype and phenotype at the human level. The other form of data that we put together with that is data from cellular systems that we generate in our own in-house wet lab. We take data that are we take cells that are derived from humans and we intervene in them also genetically using technologies such as CRISPR in order to say if we make this genetic intervention in a cell, what cellular phenotype does it cause? How does the cell change its state when we make this genetic intervention? And the reason why we like that is because unlike in the human case, here we actually get to do an experiment. On the human data side, it's the system that we want to modify, but we don't get to actually do causal experimentation. On the cellular system side, we can do causal experimentation. It's in the wrong system, but at least it's related to the right system. So the machine learning is used in our case in two ways, both to create a more meaningful representation of these high content data modalities in both humans and cells, and also to bridge between them so that you have translatable models in the lab that truly predict the effect of an intervention in a human being.
Yeah, that's a very good example of a data-driven, data-centric approach. And so one thing I would have to ask is, like, how do AI researchers in this field, like, how do you generate these large-scale, high-quality biological data needed for these AI models? Because you were talking about large-scale data way before LLMs broke onto the scene big time last October. So yeah, like, it's fascinating that you had that insight early. And then again, how do you achieve this as well?
Yeah, so I think yes, I did realize actually long before October. I mean, it was actually the beginning of my return from Coursera back into biology is when I looked around me at a point when Coursera was kind of at a good steady state and said, wow, not only is machine learning changing the world, but it's changing the world because we finally have access to enough data. And even before large language models, there was the progression that we used to see in computer vision in the early days which is when I started in computer vision, you could barely recognize objects, like a simple object in an image at an accuracy that was better than chance. And then even as 2016, you were already able to write little small captions for images saying a bear at the stream drinking water, under a tree. And so I was like, wow, that is a huge amount of progress. And the reason is because we have lots and lots of images. So how do we get lots and lots of data in biology? And 2016 was also the year when it was becoming increasingly clear that we could generate massive data sets in biology. And so where do those come from? On the human side, they come from the increasing availability of electronic health records, where more and more of that is becoming digitized. National biobanks like the UK Biobank that are capturing immense amounts of information about the humans that volunteered to participate in that study. And so that was an incredible opportunity on the human side. And on the cellular side, there is a convergence of technologies that cell biologists and bioengineers have kind of come together in the last few years to enable us to print data. So you can think of it as building an engine where as you turn the crank, it just prints more and more data about that cause-effect relationship in cell biology. So those technologies involve things like, as I mentioned, CRISPR. They involve the ability to create human-derived cellular systems from what are called pluripotent stem cells, where we can take one of our skin cell and revert it back to a stem cell status and then create a neuron or a cardiomyocyte or a hepatocyte. And so all of a sudden I can study, but with our genetics. So I can study the effect of our genetics on our neuronal development. I can measure that using super resolution microscopy, live cell imaging, whole genome transcriptional profiles, mass spec proteomics. All of those are incredibly rich data sources that were not available a decade ago. And so we're now in a world where we can collect the kinds of data that enable us to build language models for the language of biology. Now, is that large language models? No, they're kind of medium-ish language models because we don't have nearly as much data in biology as we do on images on the web. But even with medium language models, we can get a fairly long way towards unraveling patterns that a human mind simply will never be able to see.
Yeah, I just want to note for our audience, please add your questions to this conversation. Happy to take them. And yeah, so CRISPR definitely was a major breakthrough, but also just curious. So I think it was 5 or 6 years ago, just attending some of these conferences like Europe's and ICML, federated data was just coming out from academic research papers. Industry has kind of adopted it to a certain degree. It hasn't become as popular within the industry as I thought it would, given the promises. But do you feel like techniques like federated learning, where you can anonymize and sell your data and get access to these large data sets without while still protecting privacy and whatnot? You think those techniques have still a way to go, or they will help or have helped to a certain degree?
I think it's becoming increasingly clear that you can't just, ship out medical data to random servers. So I think that's something that various protections have been put in place. Federated learning is one. Even just having protected research environments, like some of those national biobanks have put in place, is a really good opportunity. It allows people to come in and do their work in a way that is, they can actually look at the data, and because looking at your data is actually really valuable in terms of building models that are truly a good fit to the data, but then have very careful controls on what goes out of that research environment. So I think there's a range of different solutions that are fit for purpose, and I think it, to my mind, it unlocks the opportunity, which I think is really critical, of letting more scientists explore biology at scale. And I think that we've seen that as soon as you unleash scientists onto a large, rich data set, magic begins to happen. People start discovering things. They ask questions that others hadn't thought to ask. They come up with research tools and methods that others hadn't developed. So creating these protected research environments, these walled gardens that allow people from all over to come and uncover new insights is, I think, really, really important. And I really hats off to organizations like the UK Biobank that have made these walled gardens accessible to pretty much any qualified researcher. And I wish more people would follow their lead, because I think it would just unleash a torrent of innovation.
Yeah, we do have a question from Michelle Gross, and I'll just read it out here. Do language models of biology strictly include DNA data, or many different and disparate types of biological data?
Actually, I would say that the more interesting language models, at least from our perspective, have been less on DNA data, where I haven't seen a tremendous amount of the use of those language models in practice. I've seen them on proteins. There's been, obviously we're all familiar with AlphaFold as a very significant landmark contribution. We do language models and imaging data in biology, so we've done incredible things with histopathology. It's those slides that those pink and purple slides that every time you go in for a biopsy get taken. So it's one of the more abundant data modalities out there, because every time you go in, when you have cancer or suspected cancer, a biopsy sample gets taken and stained and now increasingly digitized. And we find that there is an unbelievable wealth of information that is sitting there untapped, because people have not let machines interrogate those data in an unbiased way. Rather, they come in and with a preconceived hypothesis about what is important and not important. And to give a very specific example of work that I still did in my lab at Stanford, together with my PhD student Andy Beck at the time, was we took the first unbiased machine learning perspective on histopathology data. This was before deep learning, so it used more straightforward machine learning methods, but it was by design not intended to replicate the workflow of a pathologist. And basically the machine learning discovered that the most significant prognostic factors towards 5-year survival were aspects that no pathologist looked at in the context of standard of care. It was contributions of the interaction between the tumor cells and the stromal tissue that surrounds the tumor cells. This is what we call today the tumor microenvironment. And now everybody today says, of course, the tumor microenvironment, hot or cold tumors, all that stuff. We all know that today. Back in 2010, when we wrote this paper, it was completely uncharted territory. So you can argue that machine learning, in some sense, was one of the first discoverers, if you will, of the tumor microenvironment.
Oh, that's a great example. And I'm just thinking of DNA and tumors, and that makes me think of a question about treatment versus cures, right? So treating a tumor versus finding a cure for cancer. Do you find that, and obviously from my limited knowledge of biology and healthcare in general, I would assume that cures are much, much harder than treatments. And it seems like much of the industry is geared towards treatments rather than cures, because you hear about these expensive drugs coming out. At the end of the day, there's a treatment and not a cure, and all the questions associated around that. Do you feel like AI is predisposed in machine learning, because that's a great example you give, more towards treatments rather than cures, or it can help really advance cures over treatments, which is the great hope for humanity, I guess?
Okay, so boy, there's a lot wrapped up in that question. Yeah. So let me start with the whole notion of cures. Cures are just unfortunately very hard to come by in a lot of cases, because by the time the disease is diagnosed, it's often the case that the fundamental processes have been put into play, and reversing those is very, very hard. I mean, that's certainly the case in cancer. Oftentimes when it's caught, it has metastasized, spread all over the body. And so now you have to basically find something that hunts down and kills every last of one of those cancer cells. Otherwise, yes, it's going to metastasize again. And so that's just a really hard thing to do, especially because they're oftentimes quite diverse as the cancer continues to evolve. It's a genetically unstable thing. And so each of those cells is not necessarily the same. And so creating something that's a cure is very hard. I think in the last few years, there have actually been, for certain types of cancers and subsets of patients within those cancers, things that are cures. Some of the immune oncology drugs harness the fact that our immune system is actually really good at reacting to variability in pathogens, and so by unleashing the immune systems on cancer cells, you can sometimes mop up every last cancer cells and get a complete response. Other things are more limited in terms of, patient subgroups where the cancer is more homogeneous in its sort of diversity of cells within the patient. I think if you really want cures for cancer, we're going to have to get better at detecting it early before it's had a chance to become as gnarly and complex as it is. That's where the majority of actual cures are going to come by. And I think there's certainly technologies like for early detection that are coming up. I think that one of the ways in which, and by the way, all of those use AI as a fundamental tool as well, because early detection from, basically very small amounts of material that might be circulating in your blood before the tumor has had a chance to manifest in a more clinical way is also really hard and needs the kind of power of machine learning. Where I think also machine learning can help is by better understanding at an early stage of the tumor's development what patient subgroup you fall into, and then give that patient the right treatment. What's called precision oncology, I think, is a really powerful approach. And right now, oftentimes, people are treated with a one-size-fits-all medication. By the time you get to the third or fourth line treatment that might be suitable for a particular patient, you've just given the cancer yet multiple more weeks or months to continue to evolve under selective pressure to the wrong drug that you gave the patient. So I think a place where AI and machine learning can make a really big difference is in disentangling the complexity of the patient population, the heterogeneity, and getting the right drugs to the right people. I think that's much more likely to give rise to a cure. Outside of cancer, things are much more complicated because we don't even, in most cases, understand the causal processes that underlie the disease. And that's a lot of what we're trying to do, is to really get to the root causes. And hopefully that will give us drugs that are at least disease modifying. Even if they're not cures, they at least modify the course of the disease rather than just try and treat the symptoms. And I think those have been incredibly powerful when they are available. And I'll use cystic fibrosis as a great example, where we've understood enough about the mechanisms of cystic fibrosis that the current set of treatments are truly disease modifying. And you can see the night and day difference between that and something that is just trying to treat symptoms.
Yeah, incredibly insightful, and I would love to dig deeper into that. But first, just to take another audience question here from Bill Horstman, asking, would we get more progress if we unwall the garden, to your comment earlier? And that is a bigger question for me also, is like AI safety, machine learning safety is a big topic, has been for quite a while now, and increasingly important, as we've seen over the last few years and even the last few months. But what's interesting for me in this conversation is we usually talk about AI safety when AI is siloed by itself, but now we're talking about the intersection of AI and biology and drug discovery. So what measures are companies like yours taking to ensure that AI is safely used in this space? And like, is it even possible to address the potential risks and unintended consequences that may arise from, let's say, AI-driven biological research? It's a long question, I know.
Okay, and yeah, and there's also Bill's question, which is a little bit different. So let me first quickly answer Bill's question, then I'll answer your question, Seamus. So to Bill's question, I think there's walls that prevent entry of people into the research environment and walls that prevent the exit of private patient data from the garden to the outside. I think protecting patient privacy is very important, and so creating kind of walls that prevent patient data, especially when you start getting into genetic data aligned with clinical outcomes. You just want to be very thoughtful about protecting that. But one of the things, as I mentioned earlier, that organizations like the UK Biobank have done is by letting people come into the research environment and in a circumscribed, protected place, do research and across a multitude of questions. And I think that's been hugely transformative. And so those walls are the ones that I would advocate that we remove elsewhere, by letting as many people as we can come and do kind of insightful research, rather than trying to keep those data sets protected so that only a tiny group of researchers who generated the data have access to them. So I think that's a hugely valuable thing. And I think just creating more gardens would also be an incredibly valuable thing. There's just not enough high quality data sets out there that enable biomedical research.
Makes sense.
To your question, Seamus, Honestly, I think AI in the context of biomedical data to help enable new insights to emerge is one of the safest possible uses of AI. There's so many protections put in place by just the regulated nature of this industry. I mean, you can't just willy-nilly put a drug into a patient. You have to go through a regulatory approval. Whether the insight that gave rise to the drug or the patient population, whatever, came from an AI or from the brain of a scientist, I'm not sure the one that came from the AI is any less valid than the one that came from the brain of a scientist. But honestly, it doesn't matter because there's a clinical trial process with randomized case control that protects people from having random stuff that doesn't work ultimately make its way into clinical practice. So I think there's risks to the use of AI in the delivery of care, and I think there's finally a recognition that software is something that should also be interrogated, because some of the examples that we've seen of software that no one actually tested, there was no published research on, never went through peer review, and was just deployed in clinical practice, and patients were actually getting worse care than before the AI was deployed. I think those are things that people ought to be looking at.
Yeah, you make an excellent point, because as we talk about the lack of regulation for AI in general, Europe's trying to come up with, or is coming up pretty soon with new AI regulation, and AI in the biotech drug discovery, healthcare space is probably one of the most heavily regulated. And I do know friends who work for various companies around here in Boston, and even on the software side, when they're installing software into certain medical devices, there's a whole regulation approval process as well. So you do have this interaction between traditional drug discovery and software. So the regulators have had for a long time mechanisms in place for that, but you do sometimes feel that the regulators are always playing catch up. So do you have any concerns around that? Like, because it is, these regulations were built for traditional software, they were built for traditional drug discovery, and we know we're moving away from a rules-based system in traditional software to more pattern recognition. So you think it's doing well there?
I mean, if anything, I think the regulators are catching up in the sense that it's taken them a while to realize that you can't vet, or you can't approve a machine learning system in the same way that you can a static rule-based system, because the system continues to evolve as you would want it to, because it keeps getting better over time. So how do you appropriately regulate something that keeps changing is not an easy question, by the way. So I'm not faulting the regulators for not having a clear-cut answer, but at least they're asking the question now, and I applaud them for sort of thinking about this as a question that we need to resolve. No, so I don't think that in this particular corner of the world that we're working in, the unchecked, unregulated use of AI is a serious risk.
Gotcha. And speaking of serious risks, we saw the letter that came out a month ago now, and a lot of talk about large language models, a lot of opportunities for abuse there. But leaving that aside for a moment, do you think large language models and generative AI, what impact is it going to have at the intersection of drug discovery and AI? What's your initial thoughts there, or something maybe you might have been thinking about for a while now?
So actually, before we, again, there's two questions wrapped up, so I'm going to answer each of them separately. First of all, let me talk a little bit about that letter and this notion of regulating AI more broadly. There's different people who at least present the risks of these technologies in different ways. Some of them I think are legitimate. The question of what jobs will be displaced by these large language models, the question of how do we continue to ensure that if we deploy the AI, we have ways of vetting its output, given that we don't understand what's going on the inside. So how do we test for correctness of what we get out of those systems? Those are valid concerns, and I think need to be resolved. I don't know that regulation is the right way to resolve them, because I don't think that government regulators are better positioned to answer those questions than other people. I think the people who are waxing hyperbolic about, the Terminator and Skynet, I think that's we're not there. And I think if you talk to most AI researchers, they'll tell you that the thing is not conscious, it's not sentient, it's not going to go out and try and kill us. So I think that's a risk that is hugely disproportionately magnified in ways that are not supported by the technology. The 6-month moratorium, I am not convinced what it buys us, to be honest, because even if some group of organizations that are law-abiding agree to abide by the moratorium, a lot of let's just say other organizations, both within and outside the US, are not going to pause for 6 months. So we're just putting the law-abiding organizations 6 months behind everybody else, which I don't think is the right solution. So in terms of, I think I've already answered the one about the risks of.
You did. So it's fascinating to it's just so much happening such a short amount of time. I was looking at AutoGPT, which is agents for large language model, and it got more GitHub stars, which is a great vanity benchmark, in a week than PyTorch, which is a tool we've all been using for years now, in a number of years. So really fascinating. So let's just take one other audience question. If you can see from Dale Herzog there. Hi, what data points might we start collecting to build a Canaris environment descriptive library? I think that's an area I have no expertise in.
I mean, I have to think about it, to be honest. I would say that if we can start potentially aligning healthcare data with environmental data that's already being captured from environmental sensors, I think that might help. A lot of the environmental factors that drive cancer are things like particulates in the air and contaminants in the water. So you can imagine taking some of the sensor data from the EPA and aligning it with some of the electronic health records or national biobanks. And I think that could potentially be quite informative around some of the causal roots of cancer. It's a great question.
Yeah, and just to lift this conversation up a little bit as we get into our closing minutes here, you've been involved in 2 extremely influential companies, Coursera and insitro. And so what are some of the important lessons you've learned as a founder and leader in this AI-driven space, from education to biotech? And
so I learn new things at every company that I'm at. Let me just highlight a couple of the really important lessons that I've taken with me. That first of all, it's all about the people that you hire to be with you on this journey. And the culture that you put in place, which of course is a closed loop with the people, because the people make the culture, and the culture brings the next group of people and also sets up the right behaviors within the environment. And so at Coursera, I didn't know that going in. And I think we almost accidentally built a good culture because we hired such amazing mission-driven people who wanted to make the world a better place.
Nice.
At insitro, because I've now understood how important that was, I made a very deliberate effort to set a culture that is actually quite different from a lot of other cultures out there. Not to say that other cultures are not good in their own way, but we wanted to do something that was very unique and differentiated, which is to bring together people from disciplines that normally don't communicate with each other at all. Life scientists, computational scientists, they kind of talk to each other, they talk past each other because they use different jargons and they think about the world in different ways. And what we've discovered is that can be a huge risk if you don't build around that. But if you create a world in which those people are actually part of a team, they develop a shared vocabulary and a shared vision, magic just happens. And they start kind of building on each other in ways because they come from such orthogonal perspectives where the whole is so much larger than the sum of the parts. And so building an organization where that is actually the true underlying culture has been a design principle from the very beginning. And if you look at the name insitro, it is the synthesis of in silico and in vitro. So it's in our name, it's in our logo, it's in how we design project teams. All of that was because I made a very deliberate design of how to architect our culture from the very, very beginning.
That's so insightful. But very insightful how you build your teams. But let's get a little more personal here. So you have an incredible list of achievements. Like, what is Daphne's secret? What's your superpower? Like, how we've been able to achieve so much?
Well, I guess there might be two. One is that I just work insane hours, but that's something that my husband will not necessarily view as a superpower as much as a deficiency, and my children. But I think the other one, maybe more aspirational, I hope, is that I just have the ability, and maybe it's because I've worked in so many different disciplines over my life, I'm able to connect dots of things and create analogies and relationships between things that people don't often perceive as being similar to each other. And I'm able to say this thing on this side is kind of like this other thing over there that looks completely different on the surface. And that's been particularly valuable when working at the boundary of disciplines, as I have across multiple chapters in my career. And it gives you some often really unique insights, because you don't have to invent new things, because the people over here already invented one thing and the people over here already invented something else. Putting them together, it's new to both of them. And so it allows you to really leverage a lot of innovation that people that are incredibly smart have put in place. So I'm truly standing on the shoulders of giants in the work that I've done, except that I get to benefit from giants that come from different parts of the globe, if you will.
Right. Very, very well said. Fascinating. And you're giving a keynote at ODSC next Thursday, I believe. No, Wednesday.
Wednesday.
Can you just give us a little primer of your talk there?
Well, I think large parts of this interview were about what I will talk about there, because I will go into the technical details about, what are the techniques that we deploy at insitro, how do we generate data, how do we use machine learning to interrogate those data, and how that's given rise to some of our first, significant clinical insights that we're now taking into drug discovery. So I'm very excited to share that with the audience.
Yes, that's going to be excellent, and you'll be giving that keynote without me interrupting you, so even better in this discussion here. So just the last thing I have to ask is what's the best place for people to follow your work, because you're doing such great things, and you have another startup which you didn't mention in the education space, which is fascinating, and how that came out of COVID. So you're doing so much we didn't even get a chance to touch upon. So what's the best way for people to follow your work, your many works?
So LinkedIn is a place where I actually do post occasionally. I'm not as big on other social platforms, so that would be one place. And if anyone is interested specifically in the intersection of machine learning and biology and wants to make a difference to patients, then go to insitro.com and insitro.com/careers. And I think it's a great opportunity to make a very big difference in the world, while also still being at the frontiers of the field in both biotechnology and machine learning.
Very well said. So Daphne, it's been such an honor, such a pleasure. Look forward to seeing you next week, seeing you in person at some point. But thank you so much for coming on the show. You've been wonderful as always. And thank you to our audience. That's all we have time for today. And we'll see you next week. Bye.
Thank you very much, Seamus. It was a wonderful conversation. And thank you to the audience.
It's always mine.
Thank you. Bye.